about phylogenetics, biogeography, and patterns of diversification in polymorphic earless praying mantids.
Hi everyone. Yes, my name is Julio. What I'm going to present today is what is basically my PhD research. Most of the time I've been doing my PhD research has focused on helping the phylogenetic relationships of praying mantises in this particular group. So what I'm going to present today is just some preliminary insights on their biogeography, not so much about diversification, but well, I'm expecting some feedback from you if possible. So praying mantises don't really need introduction, everybody knows them. But just to make sure that we are on the same page, praying mantises are polyneopteran insects. It belongs to the most primitive lineages of contemporary insecta. They are a sister group of cockroaches. They are mostly carnivorous, actually mostly, not mostly, but all of them are carnivorous. And we know approximately 2,500 species worldwide. They are mostly distributed in tropical and subtropical habitats around the world. So now when the praying mantis word comes to your mind, the first thing that you picture is probably this one. But actually praying mantises exhibit extensive diversification in their ecological and morphological strategies. It's a fine balance between cryptic strategies and keeping the body in good shape enough to capture prey and secure their sustainment. So praying mantises mimic plants, they mimic dead leaves, sticks, moss, green leaf, dead leaves, who knows what, and you also have the typical green kind, and many other. So in terms of taxonomic character, this is great because you have plenty of variation for describing and diagnosing a high-level taxa or species and gender. However, this proved to be very problematic at the time of discovering systematic relationships among the group. So the first phylogeny that was published on praying mantids relationships came out in 2009 by Svensson and White. So this considerably changed our picture of what mantid relationships are like, because we realized that most, that 50% of the families and tribes are either para or polyphyletic, so it's basically a mess. Then the systematics and the morphology-based classification is basically meaningless. It certainly doesn't represent evolutionary relationships in the Mantoya tree of life, and can be hardly used as an empirical foundation for testing evolutionary and ecological hypotheses. So to illustrate this, I will show you this example. These are the bark mantids, the Liturgusidae. Bark mantises are species that use the vertical surfaces of trees, normally in forest and tropical areas. They share several features in their morphology, for example, a flat body, disruptive coloration, long legs, and several anatomical features in the conformation of the thorax and the forelegs. They are extremely fast. They are run up and down like lightning speeds, and they occur mostly in all biogeographic regions around the world. When you map their distribution in the phylogeny, you will see that they come out in different places. Now, this same pattern repeats itself over and over with all ecomorphs. Stick mantids show the same pattern, dead leaf mantids show the same pattern, so they are all over the place. It really is a mess. So this was bad news for taxonomists, of course, because we are completely disappointed about this classification scheme. So, and this is because of taxonomic interpretation of character was poorly done and misguided, because there was not really phylogenetic inference from which to derive hypotheses or relationships. So basically, the entire clade is plagued with homoplastic characters, which we know they are not good for systematic inference. But tragedies are also opportunities. So Project Mantoya is an NSF-funded initiative that seeks to bring order to this chaotic state of the art in praying mantis systematics. This guy over here, Gavin Svensson, is the PI of this project, and the project is based at the Cleveland Museum of Natural History. So the main goals of this initiative is bringing together all praying mantis researchers out there, which is basically myself and 4 other guys, and to solve these problems here that eventually will lead us to use praying mantises as biological model for ecological and evolutionary research. So we have to start somewhere, and in order to contribute to this project, my PhD thesis basically has 3 components. The phylogenetic systematics of polymorphic early praying mantises, which are right here. At the very is an early split in the history of Mantoria. They are earless. Hearing evolved in the sister group, so all these other guys have ears, and these ones are deaf. Also, there's an important taxonomic component over here, and what I'm going to present today is the early stages of integration of all this information to explore biogeographic trends, morphological evolution, and diversification in praying mantises. Unfortunately, this is one of the biggest drawbacks in Mantoria research, is that we are walking in the darkness when it comes to biogeography or any other major pattern. Nothing has been done, so there's plenty of room for investigation, for discovery. It's actually very exciting because patterns start popping up, and there's, hopefully something will come out from this. So these are representatives of the lineage, but you can see they are all over the place in terms of morphology. So the phylogenetic analysis has been completed, so what I'm going to show you right here quickly is the relationships of the 8 main clades that conform the earless praying mantises. Thespide is a large lineage that dominates the entire clade. It represents small species that are specialized in microhabitats. You have moss mantis here, you have sticks, you have grass, you have species living on the ground, on bark, on bromeliads, you name it. They are all over the place, very diversified in terms of ecology and morphology. Whereas the rest of Mantolia, the other 7 families, except for the 49, which are your typical green mantis with some specialized groups, most of them have very discrete morphological pattern, either stick, either bark mantises, the dead leaves, ant mantises, and this weird little guy here is single. It's only representative of the Stinophylidae family. So with this morphology, I have been able to circumscribe the whole group into 8 families, 15 subfamilies, 18 tribes, and all the genera have been revised and their morphological characters have been nailed down. But still, one of the biggest questions is why is there so much morphological diversity in Mantolia? What can we learn from the phylogeny to have an idea of what are the evolutionary forces pushing these insects into different directions? So one of the first, this is exploratory, this is very preliminary, but one of the first things to do to approach this question is exploring the distribution of praying mantises into the phylogenetic context. We try to reconstruct the ancestral states of the lineage using the phylogeny, the current distribution, and also timing the evolutionary events that lead to the present day distribution. So praying mantises basically are found in tropical and subtropical, like basically tropical rainforests over here, and different parts of the neotropical realm. And also they are found in dry and seasonal ecosystems. The greatest diversity occurs in this area, and not so much in these other areas. But when we look at we try to reconstruct ancestral habitat of the ancestral nodes using the program RASP, we notice that there are distinct patterns in the way the ancestors were distributed. Although there are some ambiguities in the nodes here, most of the distant nodes consist in combinations of dry habitats, and we can see that this is the most likely reconstruction, by the way. So we can see that the ancestors of most lineages lived in tropical humid habitats. Now, using these, I reconstruct the evolutionary history of this lineage using 4 fossil calibration points at these nodes. Basically what we obtained was that the earliest praying mantis lineage originated at some point 200 million years ago. So putting all this together into ecological context, we can see that The branches are colored according to the habitat, leading to the terminal taxa, with some shifts scattered along the phylogeny into either of these seasonal or arid ecosystems. So probably these are the few of the patterns I have been able to reconstruct so far. So basically, the ancestors of oral polymorphic yearly praying mantises, as well as all major lineages inhabited in humid tropical forests. Then, and this is very interesting, there has been at least 13 early and independent shifts from humid to dry habitats, involving mostly riparian events. This makes sense from the biological point of view because praying mantises are very habitat, they have a strong habitat fidelity. That means that genera that are found in humid forests, in tropical rainforests, for example, will be only found in tropical rainforests. Species from dry habitats or seasonal dry forests will only found these with very few exceptions. So their morphology have evolved to be strongly bonded to the habitat and the specific microhabitat they occupy within. So certainly vicariance is the most common pattern, but we also see some dispersal events in some of the dry adapted lineages. And also we see that the contemporary pattern respond to events that initiated during the upper Cretaceous. Now this is consistent with other patterns shown in different organisms. During the Cretaceous, the Pangea continent was breaking up and the Atlantic Ocean was starting to form, and that completely changed the environmental conditions in the continent. Drier habitats become more common and more widespread towards the Cenozoic. So we can see that this switching from humid to dry occur exactly within the time frame that is currently thought to have occurred with this geological time. Okay, so basically we have arid versus humid. So this is very good because these particular transitions can allow us to pinpoint specific evolutionary events in the history of polymorphic early praying mantises. So the next questions that we will be addressing with a more expanded data set is, can we detect morphological traits associated to shifts from humid to arid? Now, without looking at the data, I can clearly see some general morphological traits. For example, the brachypterism, shortening of the wings of females, is probably associated to the shift from humid to dry habitats. Also, this particular habit here, this cryptic habit that is sticking very close to your branch-dwelling mantids, occur only in dry habitats. They do not occur in moist habitats. So, but most of the variation is actually morphometric. So what I'm planning to do is modeling morphometric information along the phylogeny to see if there are trends, and those trends, if those trends can be associated with changing the diversification in these lineages associated with dry habitats. So there are really no specific conclusions about this, but rather a broader view. So this is very preliminary, so any feedback or any questions, welcome, and help me to nail down this pattern of praying mantises. If you have any questions, please, you're welcome. Acknowledgement to all the people involved, Gavin Benson, Douglas Curry, my supervisor, people from France, and collaborator at the Rwanda Finding Agency.
We have time for one quick question. I have one which is related to the last point you studied in morphology. And you have these extraordinarily cryptic forms that are adapted to different aspects of vegetation. Are there any cases in which you see species that show any remnant features of their ancestral vegetation types, for example on leaves, which are retained, let's say, in twig-dwelling mantids?
Yes, I've been asking myself the same question. I'm not at the stage of distinguishing that yet, but definitely part of my general thinking process. Yes.
Thank you very much.